Hi, and welcome back to Chapter 4. We're going to start to cover parts of the eukaryotic cell that include energy organelles and the cytoskeleton. So there are two organelles that are involved in changing energy from one form to another inside of eukaryotic cells. Those are the mitochondria and the chloroplasts. There is another one called peroxisomes that we'll touch on briefly as well. So mitochondria, those are the parts of the cell that generate ATP. Mitochondria are found in all eukaryotes. Sometimes we think, oh, the mitochondria, they're only found in animal cells and chloroplasts are found in plant cells. Nope. Mitochondria are found in all eukaryotic cells. Plants, animals, fungi, protists. Okay, now that I've said they exist in all eukaryotic cells, I'm going to tell you that there are some exceptions, but we're not going to deal with those exceptions in this lecture. We're just dealing with the basic biology stuff. So mitochondria, again, use oxygen to generate ATP. Chloroplasts, those are found in algae and plants, and they're the site of photosynthesis. And photosynthesis uses ATP and oxygen to make glucose. There are these other structures called peroxisomes, and peroxisomes, they just have a really very basic function of being involved in oxidation. We'll learn a lot about oxidation and reduction in later chapters. So again, we're focusing mostly on mitochondria and chloroplasts right now. And one of the really cool things about chloroplasts and mitochondria you might remember from earlier is that they have their own DNA. And the DNA that's found in those two organelles, the DNA is circular. And so, back in the 1960s, people were trying to figure out, well, how did these mitochondria and chloroplasts get their own DNA? And they came up with this idea called the Endosymbiont Theory. Now, the Endosymbiont Theory says that chloroplasts and mitochondria evolved from bacteria that were basically engulfed by an ancestral, a long time ago, right? An ancestral eukaryotic cell. So what is the evidence for this endosymbiont theory? Well, there is a lot of evidence, including the idea that both chloroplasts and mitochondria have two membranes, very much like the nucleus, but the inner membrane and the outer membrane of chloroplasts and mitochondria have really different functions from each other. So it seems like that outer membrane could have been like a food vacuole, and the inner membrane is the old bacterial membrane. If you look inside of these two structures, they have ribosomes, and they have lots of little bits of circular DNA. If you look at those ribosomes, the ribosomes actually are quite different from the ribosomes found in the cytoplasm of the eukaryotic cell. But remember, prokaryotic cells have circular DNA, and eukaryotic cells have that linear or non-circular DNA. Both chloroplasts and mitochondria can grow inside your cells. Well, not chloroplasts in your cells, but you get the idea. They replicate themselves independently of the rest of the cell. So they can make lots and lots of copies of themselves while your main eukaryotic cell is just sitting there, relaxing, having a good time. So we can think about the pathway which this evolution might have occurred. Here is an ancestral prokaryotic cell, and there's its plasma membrane. And over time, and this is whole ribbon of time right here, this blue arrow is the ribbon of time, that ancestral prokaryote was engulfed by an ancestral eukaryote. Now that happens over here, so stand by as we first have to form our ancestral eukaryote. And this happened as cells get bigger and the plasma membrane folds in on itself. We say that's infolding or invagination or something along those lines. It folds in on itself and here forms a nucleus, folds in on itself again and forms endoplasmic reticulum. Remember that as cells get bigger, if they keep getting bigger and bigger and bigger, the surface to volume ratio has some problems. So larger the cells are, the more membrane you need inside that cell for chemical stuff to happen very efficiently. All right, so here's our ancient prokaryotic cell that has gone through this infolding of the membrane to make nucleus and endoplasmic reticulum, and now we've got a eukaryotic cell. And then another ancestral prokaryote is engulfed by that ancestral eukaryotic cell, and eventually they evolve into mitochondria, those ancestral prokaryotic cells that are kind of living inside of the prokaryote or the eukaryotic cell. Now this happened 2 different times. The first time it happened, those things that were engulfed evolved into eventually mitochondria. The second time it happened, those things evolved into our chloroplasts. Now we know that it happened in that order because all eukaryotic cells have mitochondria, but only some have chloroplasts. So some of our cells kind of went along this pathway, like our animal cells, and didn't ever evolve those chloroplasts. And then, of course, the ones that did engulf those photosynthetic prokaryotes evolved into more like plant cells. We now widely accept endosymbiont theory as being strong science. We just have so much evidence for this, including the evidence that I've already talked to you about. We know that an early ancestor of the eukaryotic cells engulfed the non-photosynthetic prokaryotic cell that eventually formed this symbiotic relationship with its host to become the mitochondria, and that this relationship is extremely tightly linked. Without a eukaryotic cell, has a really hard time living without its mitochondria. In fact, there's some diseases associated with mitochondria that aren't functioning properly. And later, again, ancestral eukaryotic cell engulfed a photosynthetic prokaryote, and now we have chloroplasts. Sometimes we hear the word chloroplast referred to as plastid, simply because there's a couple different kinds of plastids, but we'll stick with chloroplast. So what do mitochondria do? Well, again, they're found in almost all eukaryotic cells, and they have this inner membrane. Remember, there's 2 membranes, that smooth outer membrane and this highly folded inner membrane. And these folds are called cristae, or cristae. Sometimes I'll pronounce it one of those 2 ways. And then inside of the cristae, inside of the inner highly folded membrane, is something called the mitochondrial matrix. There is some space between the inner membrane and the outer membrane, and that's called the intermembrane space. You can see that word right there, and you can also see it right here. In fact, let's go ahead and bold that. Intermembrane space. So those spaces, the matrix and the intermembrane space, there's a lot of stuff going on there, which we get like a whole chapter on, so hold on to your heels. But for now, just know that these are the structures that are found inside the mitochondria, and that those cristae, those folds, increase surface area, which of course increases the ability of the mitochondria to make ATP. Chloroplasts have the function of making glucose or sugar. And they do this by containing this pigment called chlorophyll. And we could highlight that word chlorophyll right there, as well as other enzymes that are important in photosynthesis. Again, we get a whole chapter on chloroplasts. The cells that contain chloroplasts are found in leaves and the other green organs of plants and algae. We say other organs because like some trees, like there's this tree called the Palo Verde tree. Sorry about that. There's this tree called the Palo Verde and its trunk is green. So any structure that's green contains chloroplasts. If we look at the structure of the chloroplast, there are the inner membrane and the outer membrane, right? The inner membrane is highly folded into these structures called thylakoids. They're membranous, they're stacked up one on top of another, and a whole stack of those thylakoids are called a granum. So one stack of coins is a granum, and one individual coin is like a thylakoid, if you were using money as an analogy, which kind of works. The stroma is the internal fluid inside of that chloroplast. And again, I mentioned this on the last slide, but chloroplasts are just one of many different kind of plant organelles called plastids. This is the one that we're going to be spending our time talking about. I mentioned that we would talk about peroxisomes, and peroxisomes they're interesting little structures. They are important in metabolic activity of cells, and specifically what they do here is they produce hydrogen peroxide, and then they convert that hydrogen peroxide into water. They're important in lots of different chemical reactions. Like, for example, we might talk about how there are peroxisomes in the liver, and those peroxisomes help detoxify alcohol and other harmful things that you might ingest intentionally or unintentionally. We don't know how the peroxisomes evolved. It's still up in the air, but they don't have their own DNA. So that's one important clue as to maybe how it might have happened. Next, we're going to be focusing on the cytoskeleton. And the cytoskeleton is a whole bunch of fibers that organize the structure and also different activities inside the cell. The cytoskeleton is found throughout the cytoplasm, and it actually some of it extends outside of the cell. So if we take a look at some of the functions here of the cytoskeleton, it has a lot of functions. First of all, the cytoskeleton helps to support the cell and maintain its shape. Remember that lots of different cells have lots of different shapes. You can easily tell a red blood cell from a nerve cell just by looking at it, and maintaining that shape is the job of the cytoskeleton. The cytoskeleton also provides anchorage for lots of organelles within the cell. So, a mitochondria isn't actually free floating around in that cell. It's held in place by almost like these rope-like structures that are the cytoskeleton. The cytoskeleton also can act as a superhighway inside of a cell, where we see these motor proteins, and this right here is the motor protein. You could think about this as like a vesicle, and the motor proteins will drag those vesicles along the highway inside of the cell, and that highway is the cytoskeleton. The motor proteins are so cool. They literally walk along the cytoskeleton. Let's take a closer look at that because it's so cool. Motor proteins, you can see motor proteins are going to be walking in this direction. Every time a little bit of ATP is used up, one of these legs shifts forward. So this back leg here is going to be bound to some ATP, and because it binds to some ATP, it moves to the front. And that is what it looks like with a motor protein walking along a bit of cytoskeleton. There are three important components to the cytoskeleton, three important things that make up the cytoskeleton. First is the microtubules. We'll take a close look at each of these as we go along through our slides. The microtubules are the thickest of the three. I know it says micro, but it is the biggest of the three. The microfilaments are also called actin filaments. And let's go ahead and highlight that word for you. Actin filaments. And the actin filaments, the microfilaments, those are the thinnest. So how do you know the difference between microtubules and microfilaments? The tubes are big, they're tubular, and the filaments are thin, like a filament of hair or something along those lines. They're thin. The intermediate filaments are easy because they're in the middle. They're kind of in the middle. So intermediate filaments. Here's just a really quick look at each of the three. We're just looking at size and general structure. The first one is the microtubule. You can see it does look like a tube or a straw. The actin filaments or the microfilaments, they kind of look like a braid of two hairs strung together. And then the intermediate filaments, those are interesting. Those are a whole bunch of tiny little fibrous units kind of coiled up together, like a whole bunch of little hairs coiled around each other. A closer look at microtubules here. Again, they are hollow, which is why they're so much bigger. They're tubular, right? And they're composed of this protein called tubulin. Now, the word here is protein dimer, and di means two, and mer means unit. So a tubulin dimer is made up of two subunits of tubulin. And then those are all kind of connected together in this tube shape. The function of microtubules include it's giving shape and support to the cell. It does guide the movements, interacting with those motor proteins that we talked about before. And so it's like the superhighway. And it also is important in separating chromosomes apart from each other, which we'll learn in a later chapter. The microtubules are important in the structures called centrosomes and centrioles. In animal cells, microtubules grow out from this structure called a centrosome, and the centrosome is going to be made near the nucleus. This centrosome, you get to kind of think about it as the microtubule organizing center. And again, we find centrioles in animal cells. When we do cell division in class, we'll talk about these more. Each centrosome has it's a pair, right? Two centrioles. And each centriole has 9 triplets of microtubules all arranged in this ring. So if we look at this, right, it's 1, 2, 3, 4, 5, 6, 7, 8, 9, and each is a triplet. So 1, 2, 3, 9 triplets of microtubules arranged in a ring. Microtubules are very important in cilia and flagella. So they form the structure of the things that move a cell around in space. Or in the case of some cilia, like the cilia found in your lungs, they move other objects like phlegm and mucus and bacteria around, up and out of that area, that's the delicate area of your lungs. And they do this with a beating pattern. When we get to that in class, well, we might be doing something fun with learning a little bit more about how flagella work. So again, looking here at cilia and flagella, looking at here just really quickly at the structure of them, it's just a group of microtubules, and they are sheathed by plasma membrane, so they're surrounded by a bit of plasma membrane, which you see right here. So that plasma membrane, and then you can see this is the part that comes out of the cell, and then this is the part that's in the cell. The part in the cell is called the basal body, and they have these motor proteins called dynein, which they kind of bend the flagellum and cilia and allow it to move. And you can see the dynein right in here. This is how we think that that the dynein works, and they work by kind of walking the flagella and cilia. Again, that's in quotes, walking. So the dynein on one side is going to contract, and that's going to cause that moving. And the dynein on the other side kind of releases. And then they take turns. The one on the other side contracts, and the far side releases. Contract and release, contract and release. And so that cilia and flagellum just kind of flip back and forth in space. And sort of like a propeller motion. That's a good way to imagine that. When we look at microfilaments, remember those are also called actin filaments. They are thin and solid, and they're built from these little kind of globs of actin. So actin, 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 and string them all together, and you get an actin filament or a microfilament. The role of microfilaments is to hold tension. It resists pulling forces that happen in the cell. And if you get a whole bundle of these microfilaments, that can make up something called the microvilli that are found in your intestinal cells. Let's take a look at the microfilaments inside of the microvillus. So this one of these long things here, this is one microvillus. Kind of looks like these finger-like projections that stick out into your intestines. And then inside of those, you've got this long structure of these microfilaments or these actin filaments that stretch all the way down through this base part and into the structure of your intestine. We also see that microfilaments function in moving cells because they're interacting with motor protein, a specific motor protein here called myosin. Myosin doesn't move like the stepping like I showed you before with the motor protein that walks along the superhighway, the microtubules. In this case, myosin is held in place along this myosin filament right here. And these little regions right here, they go backwards and forwards and backwards and forwards. And that's why they're called and that's why they're called motor proteins. So they move in place instead of walking along them. And so the myosin filament interacts with the actin by grabbing onto it and pulling it, and grabbing onto it and pulling it. We see this as being really important in muscle contractions, but it's also important for how white blood cells move along inside of your body, chasing down those bacteria and those nasty viruses like we know they do. Our final one is the intermediate filament, and intermediate filaments are, again, they're in the middle. They're larger than microfilaments, but smaller than microtubules. They're really not found in all cells. They're kind of found in some animals, specifically vertebrates. So you have intermediate filaments in your body. The main role of intermediate filaments is reinforcing cell shape and holding those organelles in place. They are fairly permanent elements of the cytoskeleton compared to microtubules and microfilaments, which they form and then they go away and then they form. So we're done part 3 of this, and we're moving into part 4, where we're going to learn about the stuff outside of the cell. So anything found outside of the cell we say is extracellular. Literally all that means is outside of the cell. Some of those things outside the cell actually are important connections between cells in a multicellular organism. They also help coordinate activities in multicellular organisms. In single-celled organisms, it might help with communication. So most cells do synthesize, right, and secrete materials that are outside of the plasma membrane. And lots of different functions here. So let's take a look at the first function, the first structure and function, and that is the cell wall. Cell walls are extracellular. Again, that means they're outside of the cell. What's the cell? The cell is defined as the plasma membrane. Anything inside the plasma membrane is intercellular, and anything outside is extracellular. So the cell wall is extracellular, and it is found in plant cells, but not animal cells. The cell wall protects the plant. It protects the cell. It gives it its shape. And very importantly, it helps prevent the cell from taking up too much water. We'll talk about that a lot more when we get into the next chapter. Plant cells are made of cellulose, and that's a term we've heard about before, right? We heard about that in the last chapter. Sorry about that. In the last chapter, it's made up of a whole bunch of cellulose that are embedded in with other polysaccharides and other proteins, and all together that makes up the cell wall. If you take a close look at cell walls, there's actually 3 layers. There's the primary cell wall. The primary cell wall is pretty thin. It's also pretty flexible. It's the farthest part away from the plasma membrane if you think about those 2 regions. So the primary cell wall it's hard to point with this pointer, but you can see there's the primary cell wall kind of at the tip of that arrow. Here's the cell itself, and here's this cell. So there's our very thin primary, this right there, and it's kind of reddish looking, so that helps. It's color coded with a reddish color. The secondary cell wall is much thicker, and it's not always found in all cells, but if it is there, it's found between the plasma membrane and the primary cell wall. So it's thick, and it's right up against the plasma membrane, which you can see right there, very thin. And then there's the middle lamella. Now the middle lamella is a thin layer between the primary walls of 2 separate cells. So the middle lamella is going to be between these 2 primary cell walls, right there, that dark region kind of right there in the middle. In animal cells, we don't have cell walls, but we do have a lot of stuff on the outside of our cells, and that's called the extracellular matrix. The ECM is made up of glycoproteins, which we talked about earlier in the chapter 4 lecture. An example of a glycoprotein is collagen. There's proteoglycans and fibronectin. For the purposes of this lecture, you don't need to know what they do yet. We're just kind of giving them some names, and most people have heard of collagen. The extracellular matrix, the proteins in there, bind to the surface of the cells. They bind to these structures called receptor proteins in the plasma membranes, specifically the one called integrins. And when we get into the chapter that really focuses on the plasma membrane, the next chapter, we'll learn a lot about those. We also have cell junctions found outside cells. And so there are 4 kinds of typical cell junctions. And basically what cell junctions do is connect next door neighbors, right? Cells that are right next to each other. How do they stay right next to each other? They're connected by these cell junctions. Cell junctions in both animal and plant cells help them stick together. They also help them interact and communicate because they're causing direct physical contact between those 2 cells. So the four kinds are Plasmodesmata, tight junctions, desmosomes, and gap junctions. Plasmodesmata are found in plant cells. Those are channels that perforate the cell walls. They kind of go through the plant cell walls and allow the plasma membrane of one cell to actually be in direct contact with the plasma membrane of the adjacent cell. If you look at this yellow region right here, this is the plasma membrane. And if you look at this plasmodesmata, it's just like a hole of membrane that goes right through that cell wall. Plasmodesmata, sometimes water, some small solutes can get through, but also proteins and RNA can pass from cell to cell. Remember, we talked a little bit about RNA in the last chapter. Tight junctions, desmosomes, and gap junctions are all found in animal cells. So what do they do? Well, tight junctions are kind of like zippers. They don't let things get through very much, but they do hold the cells together. Desmosomes, kind of like buttons. They hold things together, but kind of loosely. And gap junctions are more like straws, kind of like those plasmodesmata. They allow fluids to easily pass back and forth from neighboring cells. All of these are very, very common in your skin cells. Fancy word for that is epithelial cells. So just kind of heads up. If you do go on to the next class, Bio 102, you'll be learning a lot more about these. Or if you go to anatomy and physiology. One of the things that is really important to know about this lecture on cell structure is that a cell is actually greater than the sum of its parts. What this means is that all these parts of the cell don't work by themselves. They're all interacting. If you think about this macrophage that you see in front of you, right here, here's a macrophage, and a macrophage is a kind of white blood cell. Macrophages can destroy invading bacteria and invading viruses, but they do it by coordinating everything together. Like you've got the cytoskeleton, and it helps it change its shape and engulf the bacteria. You've got the lysosomes. Remember that lysosomes are part of the endomembrane system, and they're made through the nucleus and rough ER and smooth ER and Golgi and all those vesicles, right? So everything is involved there. And then, of course, the plasma membrane, which is doing the engulfing. Cell functions arise from the order that's found in cells. So function arises from order. And that is what you need to know for your cell structure test, I guess, coming up soon. I hope you enjoyed this information. I hope you found it useful, and let me know if you have any questions.